Hello and welcome to OSDR Chats. From muscle and bone loss to genetic changes caused by high carbon dioxide levels and oxidative stress, spaceflight affects us in so many ways. Today we're joined by Dr. Viktor Stolc of the NASA Ames Research Center, who will share with us insights from research on board the International Space Station that reveal spaceflight effects and their connections to diseases on Earth, as well as how the Open Science Data Repository and the analysis working groups contributed to this publication. Thanks for joining us today, Dr. Stolc. Could you please highlight the most important finding in this study?
Thank you, Jennifer, for the invitation to talk about our exciting study that we published recently this year with the title Metabolic Stress in Space, ROS, or Reactive Oxygen Species-Induced Mutations in Mice Hint at a New Path to Cancer, which I co-authored as the lead author with my colleagues from Stanford University, from SETI Institute, and the Slovak Academy of Sciences. So this was an international effort to map out what happens to mice in just less than 40 days aboard the International Space Station. And we discovered some unanticipated findings that I want to talk to you about today. So with regards to the most significant finding, we discovered a previously undescribed extremely high rate of mutation that we call hypermutation, induced in all the tissues that we studied in the mice that spent less than 40 days aboard the International Space Station. So the mice flown aboard the ISS, or International Space Station, exhibited a dramatic increase in genome-wide somatic mutation, up to 27 times higher mutation rates compared to ground controls. So we had the experiment running in parallel, simultaneously, on the International Space Station with a number of mice, and same number were housed on the ground in ambient environment on Earth with respect to gravity and ambient atmospheric conditions. And we simply compared what happens to the genome of these mice while they were flown on the International Space Station. So the mutation frequency was particularly high in actively transcribed genes. So we could tell which genes were being activated or deactivated by spaceflight. And this suggested that reactive oxygen species induced oxidative stress during the process of RNA transcription, which this played a major role in increasing the mutation frequency, unlike anyone has ever seen before. Moreover, we dissected different tissues in the mice and discovered a tissue-specific mutation frequency. In some tissues, the mutation frequency was higher, and each tissue had a distinct mutation frequency. Among the tissues, for example, eye and liver tissues displayed the highest mutation rates, while kidney and muscle tissues showed comparatively lower, but still significant mutation rates compared to the ground control mice. And we identified the unique mutations by linking them to metabolic demands and tissue-specific reactive oxygen production. So the most dominant mutation that we identified was in oxidized guanine base, which is one of the 4 bases in DNA. And guanine was the most commonly substituted base, consistent with its susceptibility to oxidation. So as I said, the oxidative burden was much higher on the space station than in the ground controls. And this we could detect quantitatively by measuring the induced mutation frequency across the genome, with higher frequency of mutations where guanine was substituted for adenine in terms of transition. And this likely caused guanine oxidation via the carbon radicals or other reactive oxygen species aboard the International Space Station. So because we compared the space station flown mice to ground controls that were separated in 2 groups, one with the same atmospheric conditions as on the International Space Station, and the other group on the ground included mice that were in ambient conditions of atmosphere that we normally breathe, that has much lower CO2 levels than on the International Space Station. We found that the elevated CO2 levels on the ISS were a contributing factor to the increased mutation load. And the study linked carbonate radicals, as I mentioned, formed from CO2 and reactive oxygen species with oxidative DNA damage, particularly the guanine oxidation. And what does this mean for the astronauts? Why is this important for NASA to consider what happens to a model system animal like mice? Well, as most people know, the biomedical research is carried out primarily in the initial stages on mammals, small mammals, particular mice, which exhibit some similarities to humans in terms of their physiology. So the links to spaceflight health risks were such that the mutations in genes were associated with critical biological functions, including muscle contraction, kidney function, liver metabolism, and ocular health, which means eye health. These findings align with known spaceflight health risks, such as muscle atrophy, kidney stone formation, liver damage, and spaceflight-associated neuroocular syndrome, or SANS, that is one of the risks of spaceflight for astronauts. So this was very encouraging that we detected the mutations to be tissue-specific with a frequency that was distinct for different tissues. And some of the mutations actually changed the coding function of the DNA into what we call non-synonymous mutations. These are mutations that, upon change in the nucleotide base, translate into through the genetic code into a protein with an altered sequence as well. And there were high frequencies of non-synonymous mutations in protein coding regions, and they have therefore the potential impact on function and health outcomes in mice as well as in astronauts if we repeat the study in astronauts, which I will touch upon later as I describe what we discovered in the mice first. So we indeed identified a very good correlation between the types of non-synonymous mutations in mice aboard the International Space Station that did not get changed in control mice on the ground in ambient conditions. And some of the genes where the mutations occurred were known genes related to cancer and hypoxia response, such as genes called TP53, MYC, and PTEN. PTEN. And these were prominent mutations in these genes that are known to cause cancer in large cohorts of populations that were studied. Over thousands of people had predominant mutations in these genes, just like the mice had mutations in those genes induced by spaceflight. So this suggests a link to cancer risk in space environments. So regarding the mechanistic insight into this oxidative stress, we discovered, as I mentioned earlier, that there is an interplay between elevated CO2 levels on the International Space Station relative to the ground, oxidative stress, and tissue-specific metabolic activity as a key driver of mutations that accumulate in space. So for example, tissues that are more metabolically active than others had higher mutation frequencies. And the rate of metabolism varies in different tissues depending on how loaded they are, for example, with mitochondria, which is the major source of reactive oxygen species. And as it turns out, the eye retina is, the eye vision cells are loaded with mitochondria and therefore produce more reactive oxygen species. And indeed, we discovered much higher frequency of mutations in the eye than in the other tissues. And the implications for human spaceflight from this study, we believe, are very significant because these findings underscore the importance of studying somatic mutations in astronauts to understand long-term health risks, including carcinogenesis and degenerative diseases. We know that the neurological system, also because of its high metabolic activity, is really susceptible to potential mutations in resulting from the interaction between the excess reactive oxygen species induced by spaceflight and the elevated carbon dioxide on the space station. So I think when we discussed this earlier, you were also curious about how the open science data repository played a critical role in enabling the research goals of this study. And
Yes.
it did so, yeah. So this was really important for us because, as I mentioned countless times before, the Open Science Data Repository, or OSDR, is kind of like a digital library where we go for data to analyze from biological model systems that are flown in space to help understand the changes in physiological changes and genetic changes now that occur from spaceflight alone. Now, we know that spaceflight differs from ground experience in that astronauts and all those model system animals experience microgravity, which is a lower gravity force in orbit than living organisms experience on the surface of the Earth. This alone, we know that gravitational gradient difference is a type of stress. In fact, stress that is metabolic in nature and induces reactive oxygen species. And here's how the OSDR data contributed. It was very easy access for us to, as I mentioned, access the digital library of all these DNA sequences that were previously not analyzed the way we analyze them. Although many different studies had been done on different tissues of animals from spaceflight, we focused specifically on the question whether or not, in addition to the metabolic deficiencies that humans, astronauts, and animals experience in spaceflight, whether it also could have induced mutation. And frankly, we did not anticipate the level of mutation frequency that we discovered. That's why we call it in the paper hypermutation, very high level of mutation. In fact, the level of mutation in spaceflight that we detected is much higher by at least 2 orders of magnitude, if I recall correctly, than lethal doses of radiation can cause to model systems on Earth. So there's something about the production of reactive oxygen species that really gets amplified in spaceflight, and this causes mutation. So OSDR provided the data in standardized formats. It enabled the computational efficiency, although, some of my team members who are bioinformaticians often say that there's more work in data prep than data analysis, right? And OSDR made that not to be true because the data were in standard formats and we didn't have to invest so much effort into data prepping the data for analysis. So that was very convenient. So I'm very grateful for that service that OSDR provides. And this enabled the computational efficiency on high performance computing systems, one of which we had utilized initially, which is the NASA Ames High Performance Supercomputing Center. And then we also used other high performance computing systems with accelerated processors to be able to digest the terabytes worth of data that we process for single DNA nucleotide changes from all these tissues and hundreds of mice. So I don't really remember how many terabytes, but maybe, hundreds of terabytes were analyzed. And if we had to data prep all of that, it would have been a bigger chore. So OSDR was very convenient for us in this regard. Of course, that also made the reproducibility and validation accuracy a given, since also other groups have used similar data from OSDR successfully. And that kind of bolstered our confidence in the results, because as I said, they were unanticipated. The level of mutation that we detected was so high that we reanalyzed the data many different times, in many different ways and different number of times to be absolutely sure that we're looking at such a high mutation frequency. Because as I said, even lethal doses of radiation have not been detected in this equivalent amount of dose to induce such mutation frequency in mice before. in studies that were carried out by other people over the past. Now, of course, when people look at our paper, we have hyperlinks in our paper to go to OSDR, to the original data sources. So this contributes to collaboration and knowledge sharing, to redirect other scientists to take a look at the same data again in other ways, to validate, verify, and that's how science is done. So OSDR makes that very easy, and it's very cost and time efficient, because OSDR eliminated the need for generating new data sets from scratch, which often is even more laborious than data preps. And this significantly reduced the time and cost required to perform this comprehensive analysis. And of course, the NASA supercomputer helped quite a bit too. So OSDR was instrumental in providing the foundational data and computational support necessary to investigate the role of spaceflight-induced oxidative stress and its impact on genomic stability, ultimately advancing our knowledge in spaceflight-related health risks. So, as we discussed before this meeting, what would be the future directions of this study? So, as I alluded to earlier in this, discovering that the International Space Station atmospheric environment contributes to induction of high frequency of mutations in mice really puts us on alert about what could be happening to the astronauts who share the same space on the International Space Station. So, as you know, the NASA Twins study provides, that had happened earlier on, provides an unparalleled opportunity to compare genetic changes in astronauts exposed to the ISS environment and their Earthbound counterparts. So we have an ongoing analysis focused on identifying whether astronauts also experience genome-wide somatic hypermutations similar to mice. But we are right now in the process of awaiting consenting from the astronauts to do the same analysis we did on mice. And hopefully we will receive that and be able to inform NASA about this potential concern for astronaut health. If confirmed, this would strengthen the case for revising International Space Station atmospheric conditions to mitigate the health risks. And I think that's very important. So what could be the potential interventions? One promising direction is reducing CO2 levels aboard the ISS, because lowering CO2 could reduce the formation of carbonate radicals in vivo in our bodies, which are implicated in guanine oxidation and mutation. And perhaps advanced atmospheric filtration systems for CO2 scrubbing technologies could be implemented to achieve this goal. Of course, there's the potential of pharmacological strategies such as antioxidant supplementation, which might counteract the oxidative stress, complementing environmental adjustments. And I think the combination of the two would be wonderful, and we hope to continue our research to help implement those if appropriate. And what would be the implications for long-duration spaceflight? Well, that's a biggie, because as we know, NASA's mission is to go beyond low Earth orbit, return to the moon, and eventually to Mars. So as we prepare for missions to Mars and beyond, understanding the interaction between environmental stressors like CO2, oxidative stress, and genomic stability is really critical, because future research will explore how modifying atmospheric composition or employing protective countermeasures can enhance astronaut health and mission success. And that's what we are about, is NASA's mission success. But I'm sure that whatever NASA discovers can also be implemented in the private spaceflight enterprise. And we're very much looking forward to also working with others in the space. And everyone can guess who those might be. So in closing notes, by enabling access to high quality data sets and fostering innovation, OSDR has significantly advanced our understanding of the biological impacts of spaceflight, and its role will remain central as we explore solutions to mitigate these challenges and ensure the safety of astronauts for long duration missions. And hopefully, I'll be talking to you yet again, perhaps even later this year, with some additional data that we are already working on.
Thank you, Dr. Stolc, for sharing your time, insights, implications, and peeks at upcoming work with us and with our viewers. It's very appreciated, as well as the mentions of your positive experiences with OSDR.
Of course, it's a pleasure to work at NASA Ames Research Center, in particular, which houses the OSDR data, and with all the folks that have put years of effort into making it available to the general scientific community. It really accelerates scientific discovery.
Thank you again. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access the data sets analyzed in this work with the links provided in the description section below. Thank you and have a wonderful day.
Thank you, Jennifer. Have a nice day.